Hello everyone. Thought I'd spend a few minutes just explaining some of the answers to the exam questions you were doing last week. Firstly, apologies for not sending your marks sooner. I did mark them reasonably early in the week and I marked them late at night and I didn't want to send anybody any notifications or anything, so I didn't send them through and then I think I just forgot about it. So sorry about that. Hopefully you'll get them today and you can look at the mark scheme and I'll try and explain it as we go through here. So if we look at the first question, we've got, obviously this is the Bowman's capsule and the glomerulus. So the answer to be straightforward, Bowman's capsule. A, we've got two blood vessels. Now if we look at the diagram that you have seen before, maybe one you've labelled, you can see that our glomerulus has been drawn like that. This one looks different, it's a different angle. And perhaps if you look at it like that, they look a little bit different, because if we look at the width or the diameter of the two vessels, they're the other way around. So which is which? Well, you learnt, when we were learning about this, you learnt this was the afferent, bringing blood into the glomerulus, and this was the efferent arteriole. So the afferent one was bringing blood in, efferent out. The afferent is the much wider one. So if we look at this diagram, that has to be the afferent blood vessel because it's the wider one. So that the name for A is the afferent arteriole. You don't know by looking where it is, you just judge by how wide the arteriole is. So that must be the afferent one, this must be the efferent one. Okay, that's the first question. On this then you had to label and name the structure, which is the effective filter. Well, you know from your notes that the filter is the basement membrane, and you know that the basement membrane is this sort of outer layer here of it's just outside the endothelium of the capillary. So this thick black line is the basement membrane. Then we've got a table here showing you the water potential in the blood plasma and the renal filtrate, and you're asked to calculate the net filtration force. So we've done an example a bit like this in our textbook or in our notes. So over here, you've got the water potential of the plasma is, well, you know that it's the solute potential plus the pressure potential. So I think everybody had worked out that the plasma is 3 kilopascals, and over here it's 7 kilopascals. So you got 1 mark simply for getting both of those right, but it asked you for the net filtration force. So blood plasma is forcing it in that direction, the filtrate is forcing it in that direction. What's the overall? So 3 minus 0.7, the difference is 2.3 kilopascals. So that's what you got for the second mark, 2.3 kilopascals. Okay, we have learnt that during ultrafiltration, we've got a high pressure in here and we've got little pores here, but we've also got the basement membrane, and the basement membrane acts as a filter and it allows small molecules like glucose and urea and amino acids and so on to pass through, but it doesn't usually allow proteins to pass through because they're too big. So that's what it says here. Healthy individuals, you don't get protein in the urine, but an indication of high blood pressure is the presence of protein. Why do people with high blood pressure end up with protein in their urine? Simply because the pressure is so high that some of the smaller protein molecules can get squeezed through and filtered into the Bowman's capsule and they don't get reabsorbed. Okay. Some of you mentioned that maybe the high pressure damaged the kidney. You would have had to say damaged the basement membrane of the kidney to get a mark. Now, I might have missed that in some people. So if you think you should have got a mark, send me a message and I'll add a mark on. It takes quite a while for me to download and to edit and so on. So it's not that easy for me just to look back quickly. So if you think I've missed out a mark, just send me a message and I'll check through. Okay, moving on to question 2. We've got a diagram here. Let me see. Okay, so obviously Parts D and E. D is the distal convoluted tubule and E is the collecting duct. Which two letters, you didn't have to do this, but you should be able to work it out now, correspond to the regions where most water is reabsorbed? Well, everybody would probably straight away think of the collecting duct, E. Where else is water reabsorbed? Well, here in the proximal convoluted tubule, water moves out by osmosis, so it's the answer to that would be B and E, but I know you didn't have to do that for this homework. Then we've got some diagrams here of the cells lining the proximal convoluted tubule. How would those cells be adapted for selective reabsorption? Well, most people talked about the fact there's lots of mitochondria, and the mitochondria provide ATP for active uptake or active transport. So you needed to give all of that, mitochondria, ATP for active transport, all those three to get 1 mark. Or the microvilli, increased surface area for reabsorption. Or you could have talked about how close the capillaries were, or the fact that there would be carrier molecules for the glucose or amino acids and so on. Over here, we've got a table, and if we look at the figures here, we're seeing, comparing the concentration of these 3 different substances with the blood in here and the filtrate at the end of here. Okay, so you're comparing with what's going in here with what's coming out of here. Why are there no proteins in the filtrate? Well, the proteins are too big to actually get filtered out of the blood in the basement, through the basement membrane. Glucose, well, the glucose does get filtered into the filtrate, but it gets actively or selectively reabsorbed in the proximal convoluted tubule, so there's none of it left in the filtrate at that point. Urea gets filtered into the Bowman's capsule, so it gets filtered in. Nothing happens to it, but the fact that there's so much water being taken out of the filtrate in the proximal convoluted tubule has the effect of causing this to become more concentrated. There's no more urea added in, it's simply water has been taken out, but there's still the same number of urea molecules, but hence the concentration goes up. So that's your explanation for those results. Part C, you didn't have to do, but we'll just chat about it now because we did mention this in last week's notes. Loop of Henle, the length of it, there's a correspondence between the degree of dryness or aridity of the environment that a mammal, such as the desert rat, inhabits. Explain the relationship. So the loop of Henle, the longer the loop of Henle, think back to the diagram, if I can see where I've put my diagram. Oh, here we go. Yeah, the longer the loop of Henle, the more and more concentrated the medulla is going to be. So the longer the loop of Henle, the more water is going to move out of the collecting duct, and therefore more water will be reabsorbed into the blood. Why is that? That's because the loop of Henle creates a high concentration of salt as you move down through the medulla. And so something like the kangaroo rat lives in a very dry or arid conditions, and therefore it needs to conserve as much water as possible so that it doesn't suffer from dehydration. All right, but again, I know I didn't ask you to do that question, so obviously I wasn't expecting you to write anything. Photograph here is an electron micrograph of the cuboidal cells that line the proximal convoluted tubule. So what are the structures? A, well, that's the microvilli, and B, well, if those are cells, that's obviously the nucleus. What is the role of A? Microvilli, they increase the surface area for the reabsorption of glucose and amino acids. Name the type. Well, they're cuboidal epithelial cells. Two differences between these cells and the cells that would line the epithelium in photograph. Oh no, sorry, I can't even read the question. So it's saying two differences between the endothelium lining the glomerular capillaries. So what are we talking about there? If we go back, this is what we're talking about. We're talking about the capillary endothelium. Now those are block diagrams. They're not an actual drawing of cells. But you know that the endothelium in the capillary are squamous, they're flattened, whereas these ones are cuboidal. So that's one difference. The ones in the blood capillary would have pores. These ones don't. These ones have microvilli. The ones lining the glomerular capillaries don't have microvilli. These ones would have lots of mitochondria. Now you can't really see that in this photograph, but you know that they have lots of mitochondria for active uptake, whereas the ones in the capillaries don't. They don't need it. So you needed to give two clear differences there. Moving on to the last question that you did, we've got a photograph of a cross-section through a kidney, and you're asked to label the regions A, B and C. Now if we go back to something I sketched out for earlier for another video, these are the regions, the cortex, the medulla and the pelvis. Some people got a wee bit confused and started labelling things like the loop of Henle and so on, but the question clearly says identify the labelled regions of the kidney. So the outer one, cortex, that has to be A. The one in here is the pelvis, and then that must mean the one in between there is the medulla. So that's the cortex, C is the medulla, and B is the pelvis. State which region A, B or C matches the descriptions. So where are most of the collecting ducts located? And again, if we go to this diagram here, collecting duct, most of the collecting duct is in here in the medulla. So the medulla is C. And where does ultrafiltration take place? Well, again, if we go back to here, ultrafiltration takes place Bowman's capsule. Bowman's capsule is in the cortex. So the answer to that is A, the cortex. Then this photograph here, it says D and E are involved in the process of ultrafiltration. So this is a sort of an unusual looking photograph. Ultrafiltration, you know, you're talking about the glomerulus and the Bowman's capsule. And this is the kind of diagram that we've looked at when we've talked about ultrafiltration. So if we look at this photograph, then we can work out, well then this must be a glomerulus, and this must be a glomerulus, and this must be one, and they're surrounded by a Bowman's capsule. So D would be the glomerulus, and then E, it's not terribly clear probably in the video, but D is pointing to that ball or network of capillaries, E is pointing to the clear bit, so E is the Bowman's capsule. Let me see, describe how the structure and function of D aids the process of ultrafiltration. So D is the glomerulus. How is its structure going to aid ultrafiltration? Well, you can think about the fact the blood is going to be at high pressure. Why is the blood at high pressure? To do with the glomerulus, well, you can say the afferent arteriole is much wider than the efferent one, and that builds up the pressure inside. Or you could have talked about the coiling creating a high hydrostatic pressure. So you get 1 mark for talking about how we get this high hydrostatic pressure. The capillaries themselves have only got a single layer of cells, and these are squamous endothelial cells, and there are pores. So that's another point. And then the basement membrane, which is the final layer around the capillary, it acts as a filter, and it allows the very small molecules to pass through. So those are ways in which the structure really enables ultrafiltration to take place. Part C, you didn't have to do, but I think we could have a look at it now. So if you're looking at your questions, I'll read it out. It says the kidney is also involved in osmoregulation. The beaver and the kangaroo rat are both rodents. Okay, so they're both rodents, rats, mice, that's what you think of. So I'll just get a wee bit of space here. Get ready to draw a few things. Okay, so beaver lives in a semi-aquatic habitat. So beaver, basically, lots of water. Kangaroo rat is desert dwelling. So very dry. So it needs to reabsorb as much water as possible. All right. Beaver doesn't need to do that because it's living, at the side of rivers and so on. It can drink as much as it wants. So there's no shortage of water there. It also says kangaroo rat produces very concentrated urine. So we'll say concentrated urine, dilute urine. Suggest and explain how the loop of Henle of the beaver would be expected to be different from that of a kangaroo rat. Well, going back to the loop of Henle, we know that it creates a more and more concentrated medulla so that more and more water can be reabsorbed out of the collecting duct. So the longer the loop of Henle, the more water gets reabsorbed. So if we think about the kangaroo rat, we need lots of water to be reabsorbed. So we would expect a long loop of Henle beside the collecting duct. Whereas the beaver, we would expect a shorter loop of Henle beside the collecting duct. We get more salty tissue there, and so water can move out. Here, we get more and more salty tissue. So water moves out, more water moves out, and more water is reabsorbed, and that goes into the blood vessels that are flowing all around there as well. So the answer basically is we would expect the loop of Henle in the beaver to be shorter than the kangaroo rat. The beaver doesn't need to reabsorb as much water because it's surrounded by water. There's a lot of water available to it. So it would have a shorter loop of Henle. Small amount of salt would be actively transported into the medulla tissue, and less water will be reabsorbed from the collecting duct by osmosis. Or I suppose you could have talked about it this way, saying the kangaroo rat will have a much longer loop of Henle so that even more salt will be moved into the medulla tissue. That means that even more water gets reabsorbed from the collecting duct by osmosis, and therefore there's a smaller volume of urine, but it's much more concentrated. Okay, hopefully that makes sense. If you've any questions, just send me a private message. And I've just realised when I've been doing that, you haven't actually seen the half of what I've been trying to show you. But sure, hopefully it worked anyway. If you have any questions, send me a private message. Okay, bye.